Now let's go over what actually happens during both glycogen synthesis as well as glycogen breakdown. So this is what glucose looks like. It has 6 carbons, which are labeled here 1 to 6. And this is what glycogen looks like. And it's just a polymer of glucose. And since we've numbered the carbons of our glucose molecule, the 1,6 and 1,4 indicate which carbons are connected. This means that most of our glucose molecules are connected in a line between the 1 and 4 positions, whereas the branches, as you can see here, are connected by the 1 and 6 position. Now as you can see, if you keep connecting the 1,4 linkages end to end, you end up with a straight line, whereas the 1,6 linkages come off at right angles or branch points. So why are we actually talking about these linkages? Well, by storing glycogen in a linear form with multiple branch points, a long polymer of glucose can be stored in a compact form. And you can see an example of this here. Now when the glucose levels drop in the body, this stored glycogen can be reconverted back to glucose, which can then be fed into glycolysis and then into the TCA cycle for ATP production. So what exactly is the difference between skeletal muscle and the liver in regards to glycogen? Well, in short, the muscle is greedy and keeps all the broken down glycogen for itself, while the liver is generous and will make glucose for the whole body. So in skeletal muscle, glycogen undergoes glycogenolysis to provide glucose and therefore ATP to be used locally in that muscle during exercise. But in the liver, glycogenolysis is not used to increase glucose locally, but to maintain the blood glucose level and therefore supply energy to the whole body. We'll now demonstrate the mechanism of glycogen synthesis by breaking down this figure from the book. So first off, we have glucose, and it's converted to glucose 6-phosphate. And where have you seen glucose 6-phosphate before? Great, it's from glycolysis, where glucose gets converted to glucose 6-phosphate and becomes trapped inside the cell. Next, glucose 6-phosphate is converted to glucose 1-phosphate by the enzyme phosphoglucomutase. And phosphoglucomutase here just simply moves the phosphate group from one spot to another on the same molecule. And if you remember, this is exactly what mutases do. They move functional groups around. Now, glucose 1-phosphate can be converted to UDP glucose with the help of the enzyme UDP glucose pyrophosphorylase. Now, UDP glucose is the substrate or the building block for glycogen synthesis, which synthesizes glycogen by adding UDP glucose to the end of the glycogen chain, as you can see here. And glycogen synthase is activated by glucose 6-phosphate. So if you have a lot of glucose 6-phosphate building blocks, you might as well store this up as glycogen. Now in our image here, we have a lot of alpha 1,4 bonds to create a nice long glycogen chain. Now the body doesn't want just one long inefficient chain of linked glucose molecules. So what can the body do to increase storage efficiency? Well, once the glucose molecule has grown to about 10 glucoses long, an enzyme called branching enzyme takes the last few glucose molecules of the chain and moves them back to a previous 6 carbon on the chain. Therefore, by adding this branch point, this glycogen chain is more efficient to store. Now, if you're following along in the book, what we just covered takes place in the first 3 steps and results in branched glycogen chains, as you can see here. Now next, we're going to start talking about breaking down glycogen. And this starts at step 4 in our figure here. And here, glycogen phosphorylase adds a phosphate group to a glucose molecule at the end of the glycogen chain, separating it from the glycogen. And here is another example of this. We see our glycogen chain and glycogen phosphorylase adds a phosphate group, separating off the glucose, creating glucose 1-phosphate. And now while we're on it, where have we seen glycogen phosphorylase before? Well, this is the same glycogen phosphorylase that we talked about, which regulates glycogenolysis, along with the enzyme glycogen phosphorylase kinase. Now, in glycogen breakdown, as we're talking about currently, glycogen phosphorylase can subsequently break down alpha 1,4 linkages in the glycogen chain until it gets to a length of about 4 glucoses. And this 4 glucose structure is called limit dextran, and you can see an example of this here. Now, as we just stated, glycogen phosphorylase cannot break this down any further. So to address this, 4-alpha-D glucanotransferase, which is a part of debranching enzyme, is used to move 3 molecules of glucose 1-phosphate from the branch to the end of the chain. Then, after this, alpha-1,6-glucosidase, which is just another part of debranching enzyme, swoops in to cleave off the last molecule of glucose 1-phosphate from the chain. And now, thanks to debranching enzyme, our chain is now rearranged, which allows for glycogen phosphorylase to resume its function of chopping down this long glycogen chain. Now, in a smaller reaction, glycogen can also be degraded in lysosomes, and this requires the enzyme lysosomal alpha-1,4-glucosidase, which is also known as acid maltase. And as the name suggests, it can only break down the alpha-1,4 linkages in the glycogen chain, slowly converting glycogen into free glucose. Now, also in this figure, you can see where the defective enzymes of the glycogen storage diseases exist, with glucose 6-phosphatase being deficient in type 1 glycogen storage disease, or von Gerke's disease. And in type 2 glycogen storage disease, or Pompe disease, you have an acid maltase deficiency. And in type 3 glycogen storage disease, or Cori disease, the alpha-1,6-glucosidase function of debranching enzyme is deficient. And in type 5 glycogen storage disease, or McArdle disease, skeletal muscle glycogen phosphorylase is deficient. Alright, now let's take a shot at a flash quiz. What are the end products of glycogenolysis? Now, we just covered how glycogen gets broken down, but the end products of glycogenolysis in the liver are glucose as well as a free phosphate. But if you remember from earlier, muscles are greedy, and they keep all the broken down glycogen for themselves. Therefore, muscle breaks down glycogen into glucose 6-phosphate, which can enter glycolysis within the muscle to make some ATP. Well, alright, I guess that'll wrap up our review of glycogen. See you in the next video.